If you don't mind, I'm going to get started. We have a lot to cover today, and I wanted to go back and review a couple of really key things before getting into today's section. So instead of talking to you about a Nobel Prize that's already been awarded, I wanted to give you a little bit of a flavor of a potential Nobel Prize. It keeps getting mentioned every single year that Carl Dijsselroth and Ed Boyden might actually, sorry, Shalini, can you just move that one? No, your bag a little to the, thank you, sorry. In terms of winning a Nobel Prize, I'm going to present to you a field known as optogenetics, and it's actually a really powerful tool. You'll need to know it because you'll be designing an experiment on the final exam. This year, we're going to try practicing it several times in sort of an online environment. If you're not comfortable with molecular biology nor molecular genetics, we'll make sure that you are comfortable by the time you get to the final exam. But there are some key points that I need to point out to you in today's lecture, but I wanted to give you an idea of why we're looking at this. It's a very, very powerful technique developed and published first in 2009, and it's since been almost a standard in various neurobiology labs. So we'll use depression more as a tool to understand a lot of biological processes rather than studying it as a biological phenomenon. So I'm going to go back, review a little bit about depression and a little bit about some of the putative targets. We talked about two different genes that were mentioned by a couple of individuals, including Eric Nestler, who was looking at delta phos-B. We're going to revisit Eric Nestler's work again today. And in addition to Eric Nestler, we had also looked at the fact that instead of genes going down, there was a gene that was actually being increased, MKP1, and that makes it unusual. In a process like depression, if you have something going up, it actually is an indicator that something actively is being expressed that might be leading to depression. And certainly everything that that reverses that trend shows that you can reverse or block depression. But that's really where we're at right now. And I mentioned one last model that I'll get to in a few slides. I sort of stopped at the end of the last lecture on neurogenesis, and neurogenesis is also a really huge field, and we'll be seeing neurogenesis several times before the end of this course, because in your neurobiological backgrounds, whether you take another course in fourth year specifically on neurogenesis, like CSB 430, or you take other courses that are related to neurogenesis, it's an area that is undergoing a lot of investigation, not just for neurodegeneration, but also in terms of cognition, and other factors as well. And we'll touch upon that briefly today, and we'll come back to it several times in this course. And of course, because it's the molecular cellular basis for a lot of different behaviors, we have to look and examine some of the molecules that are associated with neurogenesis and the brain regions that are involved in neurogenesis in the adult. And again, putting this back in the context of what might happen in depression. The flip side of all of this is that we don't understand this process at all, but we have a biological description of what the opposite of depression might look like. Some of you might undergo stressful situations, whether it is something that you do to yourself, so think back to the experiment that I did on myself, or it might be something related to a condition completely out of your control, out of your hands, and you have a stressful condition, or maybe you'll have a series of stressful conditions, and how individuals handle and respond to that stress biologically can either lead to depression or not depression. And we call that condition resilience. And we know that that biological factor of resilience actually exists. We know that mice can be bred for resilience, and we think that there is a biological basis that involves the firing between the VTA and nucleus accumbens, and I'll get into the overview of that, and this gets into optogenetics. You would not have been able to understand resilience, or at least start to tease apart resilience without using optogenetic methods that I'll be going through today, and I'll begin with that in a few moments. So I had talked to you about this slide, and this slide is just a review for you. I've actually given you this slide twice. You'll see it again a little bit later on. But this slide is important in the sense that this model here, the social defeat model, where you take a larger animal and you put it in the cage with a smaller animal, this smaller animal will feel like it's being bullied. It will exhibit social defeat. And this animal here, well, if you keep doing this on a chronic basis, will have changes to its brain. And one of the things that I'll show you a little bit later on is the change that occurs in the firing rate. If there is an increase in the firing rate between the VTA and nucleus accumbens, you are going to be more and more susceptible in this social defeat setting to developing depression. And we're going to be looking at that. In the last lecture, one of the things I told you was that in different areas of the brain, delta phos B expression often is an indicator of whether or not depression might occur in different models like learned helplessness. And it's very important that you separate these two out. Why they're not universally the same isn't really well understood right now, but many researchers, including Eric Nestler, have tried to identify the potential reasons for this. So we talked very quickly about inducing depression in these different models, whether it's chronic unpredictable stress, the best model that's available to us, because it looks like what happens to you in real life. You are not expecting to have stress applied on you. Well, maybe you are. If you're in U of T, then it's unavoidable that you'll have stress, especially like right before reading week and right before final exams, right? And overall, though, in life, it is unusual for you to be able to tell when stress is coming. So when you have this unpredictable form of stress, it's actually a really good model that mirrors in many ways how life really works. And we talked about some of these and how we can justify whether or not depression has actually occurred in these different models, like chronic unpredictable stress, in social defeat, and in others. And again, this gets into this idea that most of our models right now are not genetic in origin. These are induced models. We do not knock out a gene and induce depression. We can look at the induction of depression by using these different models like chronic unpredictable stress or social defeat models. So I told you also that because of that, because we don't have a reliable model for depression, that one of the things that everyone started to look for were these different genes. If we can identify a gene or a series of genes maybe, and we could modify or manipulate the expression of those different genes, then it might be possible to actually have a better biological basis for what depression looks like. And I told you that one of the biggest studies that came out was a prospective study of individuals in New Zealand. And this is the study that was done back in 2003. And this was a prospective study. I don't know if did I speak to your class about prospective studies and what those represent? Can you guess what a prospective study is? Just by its name. Yes, Ashkin. So you're analyzing a cohort for a long time. These are, by definition, incredibly difficult. So imagine today I have a hypothesis in 2015. In 2015, I'm going to say that 10 years worth of stress, or if I look 10 years from now and I look at what the effect of stress is 10 years from now, I'm going to hypothesize that all of these different things will happen. And this is what a prospective study is. It's a long-ranging study of cohorts. And this is what was done in that study that I talked to you about in 2003. And it was a very involved study. It was a very carefully controlled study. And at that time, one of the things that came out was that if you had the long version of the 5-HTT allele, and you had 2 copies of that, you were less likely to develop depression if you encountered stressful situations. And if you carried the short alleles of the 5-HT or the serotonin transporter, you were more likely to develop depression if you were exposed to stress. And that was, again, big news because a potential target gene had been identified. Unfortunately, when you go in and you try to manipulate this gene and you look for an animal model for depression, it doesn't seem to work very well. However, overall, it did give us a good idea about how different genes fit into this story of neurotransmitter decline and how these different declines in neurotransmitters might lead to things like depression. Because if you go back, there is no doubt whatsoever that if you are depressed, your neurotransmitter levels actually do go down. However, the inverse of that, if you take an antidepressant medication that's supposed to be helping you and your neurotransmitter levels go up, you don't feel any better right away. And that was sort of the conundrum. So yes, it's great to talk about all of these different neurotransmitter levels. Yes, dopamine levels go down. Yes, serotonin levels go down. other neurotransmitters also go down, like norepinephrine and noradrenaline. This accounts, or potentially could account for some of that if there's a deficit in the total number, if you have the S allele, the total number of the serotonin transporters, and if it takes a while for you to actually form serotonin over time, especially in stressful situations, you might not have serotonin around in order to be able to accommodate for any stress-induced change. And this is sort of the last slide that we left off with, because the neurotransmitter hypothesis, the one that has dominated neurobiology for the last 25 to 30 years, is actually gaping with these big questions that have not yet been resolved. And one of those is, why don't antidepressants, if they increase your neurotransmitter levels within an hour to 6 hours after you take it, and you can measure that on a PET scan, you can see that right away, why doesn't it actually affect the mood of an individual? And that has become the biggest question. Why does it take time, in other words, for antidepressant medication to actually be effective? And this gets into this other theory about neurogenesis and the role of different neurotropic factors. And so I mentioned at the very end that in your brain, in the adult brain, even though it wasn't believed for a very, very long period of time, and Fred Gage down in UCSD, and I think at the time he was at La Jolla, he had actually proposed that in the adult brain, not during early development, not after you're born, but in the adult brain, all of your brains right now, you have neurogenesis going on. And of course, no one at the time believed him that neurogenesis could potentially occur in the adult brain because the circuits were supposed to be locked in place. Why would you want to have new neurons if your circuits are already locked in place? All of the neurons have found where they need to go. Why would you have to have this? However, over time, it became more and more obvious that there were two places in the brain that actually had regions that could actually undergo adult forms of neurogenesis. One of these being the subgranular zone as part of the hippocampus. And this is where we're going to begin our understanding of neurogenesis and its potential role in depression. And the other one being the subventricular zone. And I'll get to that a little bit later on. We think that there is a defined process by which a neural progenitor cell within the hippocampus that is normally found in the subgranular zone, they start to proliferate because that's what stem cells do. And as they proliferate, some of these will survive and they will start to differentiate and become neurons. And these neurons are believed to be really important, especially in depression and in other biological phenomenon. So this is a really busy looking slide, and there are a couple of things I'm going to highlight for you that you should make sure that you are comfortable with and understand. So one of these is that we have a resident population of stem cells. We'll just call them stem cells, and I'll get into the nomenclature in different areas of the brain in a few slides. But we have these stem cells that begin, and these will form other types of neuronal-like progenitor cells, and we'll call these neuroblasts. And these are found in this area of the hippocampus, in this part of the hippocampus, in that subgranular zone layer. And when they start proliferating, and to the time that they actually fully integrate into the hippocampal circuit, it takes somewhere between 3 to 4 weeks. Down here on this particular diagram, it shows you about 2 to 4 weeks. Why was that really interesting for neurobiologists? This is about the length of time that it takes after you have started an antidepressant therapy. And if that antidepressant therapy works, there's no guarantee, by the way, that an antidepressant therapy will work on every individual. It's only about 40% to 50% efficacy. You get drug A, you try it for 3 to 4 weeks, and if you feel better, you stay on drug A, and if it doesn't work, you're put on to drug B. But it does take time for that drug to make you feel better, that antidepressant medication. And it takes about the same length of time as neurogenesis takes to occur. Now, there are a couple of important molecules to keep in mind here. So the overall stages from the time that this stem cell has decided to become a neuronal-like lineage, or decides that it wants to go on the road to becoming a neuron, when it starts proliferating in this subgranular zone layer in the hippocampus, we can actually mark this proliferation, and we can mark the fact that it is this early stem cell by two different proteins that are found uniquely in these types of cells, which are GFAP as well as nestin. And these are markers that you'll see disappear as they start to mature. So after they start to proliferate, divide, and as they start to become more and more neuronal-like, they lose these markers like GFAP and nestin, and then they start to express other markers like NCAM as well as double cortin that I'll show you in a while. And all of these things, as they start migrating and as they start making axonal targets and meeting our axonal targets, they'll start to express other markers. These markers are really important because we're going to see these markers several times today. So one of these markers is nu-N. And at the very end of today's talk, I'm going to show you a marker for neurons. So if you're ever asked, if you have to have an antibody and you're not sure, you have a whole collection of cells in your dish, or you have a whole collection of cells on a histological slide, and you only want to stain neurons, what antibody do you use? You use nu-N, because nu-N is a marker of mature differentiated neurons. And it doesn't matter what kind of neuron it is. It could be an inhibitory neuron, it could be excitatory, it just marks neurons. In addition to that, we'll also see as these neurons start to integrate into a synaptic circuit, and as they start to mature, that they will also express another molecule known as calbindin. And sometimes, if you're reading different papers, you'll see this as calbindin D28K, which tells you the overall size. But I want you to make sure that you know the markers that mark proliferation versus markers that might be in neurons that have started to differentiate, and as they fully differentiate, that we can mark them with totally different types of markers like nu-N as well as calbindin. And you'll see that there is a little bit of overlap depending on the maturity of these different cells as they start going from a full stem cell into a neuronal-like neoblast, and as they start forming more and more neuronal properties until they eventually integrate fully into the hippocampus. And this, again, is where we find it in the subgranular zone layer in the hippocampus. So why are we talking about this phenomenon when we're talking about depression? It had been known for a very, very long time. Actually, around the same time as neurogenesis was starting to become really popular, it had been known for a while, since at least 1997, when these studies had become replicated several times, that there is actually a decrease in neurogenesis levels within the hippocampus. So about 5 years after Fred Gage had sort of indicated and had shown, and everyone else was able to replicate his results, that neurogenesis does occur, when people looked in 1997 that what happens in the brains of these depressed animal models, the induced models, they saw a decrease in the levels of neurogenesis. Going back to an earlier slide, in an earlier lecture, do you remember anything about depression and the hippocampus, or anything related behaviorally to depression and the hippocampus? So does the hippocampus get bigger if you're depressed? So one of the things that we had talked about, that ultrastructurally as well as biologically, the hippocampus tends to shrink. And one of the reasons why this was examined was maybe if you affect neurogenesis, that might account for the shrinkage in the size of the hippocampus. Do you have problems with your memory and cognition if you have depression? Yes, you do. And again, this is another reason behaviorally why individuals were looking at this area of the brain. In addition to that, one of the other things that makes animal models really impressive is that if it really is a good model, it should recapitulate some of the things that we see in humans. If you were to take those depressed mice, the induced depressed mice, and you were to start treating them with fluoxetine or Prozac, a typical antidepressant, when you start doing that, they actually will get better. They will stop exhibiting the signs of depression. And by the way, how do we measure that behaviorally? How can we see if an animal stops being depressed? Can you tell me anything that you're familiar with? Yes. Could be food intake. What's another way to measure anhedonia? Yes. Yeah, sucrose preference. So you can measure that. If they go back and they start liking sucrose again, then that's one way to see if your antidepressant is reversing this depression. And they do. They go back. They like the sugar water again. You might test them on a tail suspension test. If they haven't been struggling before and they just sort of hang upside down and they don't struggle to upright themselves if they're depressed, after they're given fluoxetine and Prozac for a couple of weeks, they will go back to struggling, trying to uh, rewrite themselves. Still, in these animal models, it took time for this process to occur. It takes, even in mice, time for this process to occur. Later on, other studies, again using meta-analysis, have we're also able to put another link into this chain as well. And one of the things that is now known is that whether you're taking antidepressant medication or on antidepressant therapy, ADT, you'll see it either as ADT for antidepressant therapy or antidepressant medication, ADM. One of the things that happens is if you are on these, you will increase your levels of BDNF. BDNF, again, is something we've talked about and is very important in the health of neurons. And in addition to that, you will increase the level of neurotransmitters like norepinephrine and serotonin. Stress, on the other hand, is actually really bad in the hippocampus. In these CA3 pyramidal cells, it will decrease the levels of BDNF. And overall, one of the things that these hippocampal neurons look like is that they look shrunken, and the dendritic arborization, the number of projections they send out, are dramatically atrophied. Whereas with antidepressant therapy with increased levels of BDNF, we have increased cell survival as well as increased growth. Now imagine you are a newly born neuron. You are a progenitor cell. You are a stem cell in this context where you don't have trophic support. You don't have growth factors around. You don't have the appropriate signals around. The most logical consequence of that is you're not going to survive well. And that's precisely the reason why in stressful conditions, young neurons don't tend to survive. There's not enough trophic support, and the stress is actually deleterious to those neurons. In addition, why is this whole process? Why does it involve things like dopamine? Why does it involve things like monoamines, like serotonin and others? One of the things that seems to be really important out of all of the different types of neurotransmitters, so glutamate overall isn't affected. GABA isn't overall affected in terms of the neurotransmitter levels in your brain. But why do these neuromodulators like dopamine, serotonin, norepinephrine, or noradrenaline, why are they affected and why are they so important? There seems to be some suggestion that these particular types of neurotransmitters or neuromodulators actually are things that will prime the BDNF expression as well as release. So if you're having a drop in these monoamines, it means that as a consequence, you're going to have a drop in these BDNF levels. And if you have a drop in BDNF, you're going to affect neuronal survival of the ones you already have there. But in addition, you're going to affect the neuronal survival, pathfinding, and everything else of those newly born stem cells that you have in your hippocampus as well. And again, if you go back, and the big name in this field right now is Eric Nestler. He's not the only person that's been looking at this, but Eric Nestler seems to be a big player in the field of motivation, depression, as well as addiction studies. And again, you don't have to worry about all of the details down here. It's really recapitulating what I've just told you about the effects of different monoamines on cell survival. So again, why is all of this important? Why do we need BDNF? Just as a refresher for you, if we have things like BDNF or NGF, and we'll concentrate on BDNF because it's the one that seems to be the most important overall in the brain. If we don't have BDNF, if you've stressed out your neurons, you've decreased the levels of norepinephrine, serotonin, and dopamine in various areas of the brain, you don't have BDNF. If you don't have BDNF, all of these downstream signals in every neuron that normally would respond to it, including progenitor cells, would no longer be able to turn on any of these three different pathways that we have talked about previously. So one of those pathways that was really important was the activation of this AKT pathway, or the PI3 kinase AKT pathway, that turned off this apoptotic pathway that is here. If we don't have AKT, this pathway now dominates, and this cell is committed to undergoing apoptosis. in a very coordinated fashion. And again, all of this leads to things that get transcribed at the level of the nucleus, things like Krebs. And Krebs, in addition, causes the cell survival, differentiation, and neuritogenesis. So imagine if none of these pathways exist, whether in the hippocampus, and they start to atrophy as a result of none of these processes being turned on, and this process being turned on, sorry, none of these processes are activated, and this process becomes activated. So we don't have neuritogenesis, which leads to atrophy and the loss of the dendritic arborization, no more cell survival, and no more differentiation. And you might think, neural differentiation, these are already differentiated neurons, right? Absolutely. But imagine now you are a progenitor cell, and you no longer have the signals that allow you to differentiate. So this, again, is one of the reasons why this particular process becomes so important. This is a really busy slide. I'm not going to go through this in a lot of detail. There are a couple of things that I want you to know. Since that time in the late 1990s, we've started to tease apart a number of the other molecules that seem to be important. I told you that overall BDNF in your brain is one of the key players, and that's universally true. If you want a healthy brain, you need high levels of BDNF, and you need the right isoform of BDNF, but overall having any BDNF is advantageous for your brain. We know that through a number of processes that we'll come back to later at the end of March, that stress overall leads to a decrease in the production of various neurotropic factors like VEGF as well as BDNF. We know that antidepressants, if you are taking them because you are not feeling so great, you have a mood disorder, you have depression, will actually increase the levels of different monoamines, which in turn will increase the levels of different neurotropic factors like BDNF as well as VEGF, insulin-like growth factor 1 in the brain, as well as fibroblast growth factor 2 or basic fibroblast growth factor. All of these things work through the same pathways that I showed you on that previous diagram to turn on 3 different pathways that are good for cell survival, differentiation, as well as growth. And that overall, if these are turned on, it will lead to having a really healthy looking neuron. If, on the other hand, stress is coming along and causing a reduction in things like VEGF and BDNF, you will start to have atrophy of spines, atrophy of dendrites, your neurons start to shrink. Overall, that gets affected on a macro level by having the hippocampus start to shrink as well. Ultimately, behaviorally, not just in the hippocampus, but in other parts of the brain, and we have talked about other parts of the brain being affected, this might lead to depression as well as some other behavioral consequences down the road. Now, we know a couple of other things in addition to all of this. So I'll ask you to make sure that you know that VEGF, for example, and BDNF, especially in the cortex and in the hippocampus, actually has 2 different effects. And this is sometimes forgotten in the overall scheme of things. You keep hearing like, you know, BDNF is really good for you. It's great for your hippocampus. It is good for your hippocampus, but maybe not in the way that you might have thought of it previously. There are 2 growth factors that actually are really important in the hippocampus and in the cortex. And these 2 different growth factors are the vascular endothelial growth factor, or VEGF. And one of the things that VEGF does is when you have a large amount of VEGF, it actually increases the rate of proliferation. And I told you that in the subgranular zone layer in the hippocampus, we have cells that proliferate. So if you have VEGF, it increases the rate of proliferation. If you have stress, you decrease VEGF, you decrease the rate of proliferation, which is a drawback to what is going on in the hippocampus. But VEGF is only part of the story. If you want those cells that have proliferated to actually survive, it's like going out and creating all of these cells, but every last one of them dies. That's of no advantage to you. And one of the other things that has to work in concert with that, and which is why BDNF is so important, in these newly formed neurons, in these neoblasts or neuroblasts, BDNF will increase the survival of those proliferating newly born neuroblasts that have just formed. So BDNF is important for that neuronal survival, not just in the adult neurons, but in the ones that are forming. So we need both in order to have the proper formation of these different neurons. So I wanted to take a little bit of a sidetrack here and talk to you a little bit about these two different regions in the hippocampus, and then talk a little bit about the putative roles of these different regions overall in the hippocampus. I told you that in the dentate gyrus, especially in the subgranular zone layer of the dentate gyrus, we have a group of neurons, a group of stem cells that are there that overall maintain the size as well as the number of neurons that are found in this structure known as the hippocampus. So they start off here, they start off in the subgranular zone layer here, and they will start proliferating, and it takes about 2 to 4 weeks after they've started dividing to eventually form mature neurons that integrate fully into the synaptic context of the hippocampus. If you were to take a look though, and we don't talk about this in a lot of detail in neurobiology for whatever reasons, there's actually a structural difference in terms of the hippocampus, both in terms of the dentate gyrus and the CA1, CA3 layer. But in addition to that, for whatever reasons, within the hippocampus, there are actually functionally different types of receptors. And we're not really sure of the reasons why this exists. But we know, for example, that if you look at the inhibitory receptors, the ones that respond to GABA, and these are inhibitory receptors, within the dentate gyrus, all the neurons in the dentate gyrus, including the ones that are newly born, will eventually express, if they stay in the dentate gyrus, these delta subunit containing GABA A receptors. These are very different fundamentally than the ones that are found in CA1 and CA3, where they have another form of the GABA A receptor, which is known as the alpha 5 subunit containing GABA A receptors. For those of you who have taken other neurobiology courses, or you might remember from your second year class, what's the difference between these two? Or what's the similarity between these two different types of GABA A receptors? Do you know anything about GABA A? Yes. It is an inhibitory receptor. Early in development, though, at this stage, it's actually neither inhibitory nor is it excitatory. It's sort of neither. But these are actually special types of GABA A receptors found only in this area of the brain, as well as in the olfactory bulb, which hopefully you'll see why is important in a few moments. Anything else that you know about these particular subunits of GABA? Yes. They are all ionotropic. You're absolutely right. So they conduct very quickly. Anything else that you know about these? So there are actually two different types of receptors generally. One is they're synaptic, and the other is that they're extrasynaptic. They're not immediately found across from the synaptic complex. They're not found immediately opposite the vesicles. Are these subunit containing GABA A receptors synaptic or extrasynaptic? 50-50 chance. Extrasynaptic? So for those of you who are saying extrasynaptic, you're absolutely right. So this makes these neurons actually really important in our study. And these are ones that I used to study a while ago when I was a postdoc here at U of T, in terms of why these different stem cells have these different classes of extrasynaptic GABA A receptor subunits, and how we can actually identify where they're coming from. So if you have neurons that contain delta subunits, we know that they originated from the dentate gyrus. And if you have other neurons that contain alpha 5, it's a marker that allows us to separate out these different classes of neurons. So again, one of the few recognized sites for this adult form or postnatal form of neurogenesis. And the other one being the subventricular zone. So we've seen this diagram before, and I'm going to go through this again for a couple of different reasons, just to make sure that you know the different molecules that are involved in this whole process. So this bright blue band down here that you see in this photomicrograph, every single one of these is actually one nuclei. Okay, so you can't see them because they're so densely packed, it looks like it's a line. But in fact, each of these represents like one single nucleus. And it would look a lot like this up at the top, but all you see is this bright blue line. And what it's being stained for is DAPI. And DAPI is a fluorescent molecule that stains any cell type. Doesn't matter what it is. Could be a neuron, could be a glia, could be any cell type, could be a fibroblast, could be an endothelial cell. As long as it has DNA and it has a nucleus, DAPI will stain it. And it stains all nuclei fluorescent blue. So you can see that there are many, many, many nuclei down here. And what you can see being shown here is that in green, these are the adult-born neurons that are not ubiquitous. You don't see them everywhere within the hippocampus. You only see them here in this subgranular zone layer of the dentate gyrus. So as they go from this stage, at stage 0, all the way up to about 4 weeks of time, what are the different types of markers that we can look at in some more detail? Again, there are a couple of things, and very, very controversial, by the way. This is a great time to be a neurobiologist in this field because there's so much controversy and there's so much interest in this. We know, for example, that young adult-born neurons, I know it sounds a bit like a mistake, but if you're younger, and you're still considered an adult, these younger adult-born neurons actually generate much more enhanced excitability. They're much more likely to start firing. Once they are locked in, they don't seem to have as much plasticity as they will later on once their circuits are fully formed. At this stage, as they're just starting to get formed, they will have much more plasticity. I told you before that remember those diagrams where we looked at LTP and LTP only went up to like 150? In these neurons, you can actually jump way past that. We don't really understand that either. They're much more plastic. They're much more malleable than the ones that we have that are parts of these integrated circuits. They have much more excitability. They have much more synaptic plasticity than other ones that you might encounter in the adult. So these adult-born neurons are important to us because they get readily activated by a number of different learning experiences. Because they're so plastic, these newly integrating neurons are really important for us. And they might be really important in certain forms of memory performance. This, again, is a really big source of controversy. Some of you who have taken psychology courses might have heard of Paul Franklin. Have any of you read a paper by Paul Franklin recently? So one of the things that he proposes is that you can't remember things past the age of 3. If you've ever thought of it, can you go all the way back in time to when you were 2 years old? And can you remember things from when you were 2 years old? Can anyone do that? Everyone's trying really hard, though. That's good. I like that. Thanks. One of the reasons why he believes that that's true is that during year 2 to 3, you have this massive increase in neurogenesis that will eventually populate your brain. Those are the ones that are actually much more synaptic. They have much more synaptic plasticity. And if they have much more synaptic plasticity than the synapses that already exist in your brain, what they'll do is they'll outcompete them. And you have no memories because those memories were outcompeted. Those synapses were outcompeted by these newly born young adult-born neurons that were much more synaptic plasticable, I guess. Okay, so that makes it a really interesting phenomenon to study in terms of psychology as well as in neurobiology. So the other thing that is also universally true, and if you're in Dr. Moorshead's neuroanatomy class, you should ask her about it. One of the things that she does in her lab is she actually studies neurogenesis, but more importantly, neurogenesis and how it changes both in disease as well as in aging. And unfortunately, as you likely know, as you get older, not only does your brain start to undergo a little bit of atrophy, but in addition to that, neurogenesis in the various areas of the brain also declines with age. So I have a lot less neurogenesis going on than in the brains of all of you. So there are fewer numbers of progenitor cells. The progenitor cells I do have, they're not dividing as quickly, nor are they surviving as efficaciously. And so it has some implications for different neurodegenerative diseases. Is this one of the things that we could reverse, for example, in Alzheimer's disease? Might it be important in things like schizophrenia? And of course, we're looking at this through the lens of depression and stress-mediated anxiety and other types of disorders. So enhancing neurogenesis in all of these conditions might be really important for all of us if we're looking at how this might play a role, not just in memory, but in other types of behaviors as well. Does Dr. Moorehead, by the way, talk about her research at all in her neuroanatomy lab? Not at all. Oh, that's a shame. She has some great research, so hopefully you'll have the opportunity to ask her about it. Do you have a question? So why might we be more susceptible to depression if we? Why aren't we? Actually, that's a really good question. So this gets more into psychology. So I'm going to take a little bit of a side track. As you get older, and if you have the opportunity, you should definitely take a course in positive psychology. Forget who teaches that now. Dan Dolderman teaches that. And as you get older, one of the things that happens is you learn to adapt to things a little bit better. You have a more positive outcome on things. Mind you, there's one aspect that isn't covered in positive psychology that's equally important. If you fall into depression later, it's not within like the 15, 20-year range, but if you fall into depression when you're 30 to 40, and you're a male, you are much, much more likely to commit suicide than if you fell into depression earlier. So there's that aspect of, yes, you're less likely to have depression, might be related to neurogenesis, or you might have better coping mechanisms to deal with stressful situations or view things differently in terms of stress. That's pure psychology. In terms of some of the biological outcomes, we do know that individuals, especially males that are older that have depression later on in life, they're more likely to commit suicide than individuals who are younger. And there's this range, like teenagers also had that same propensity, maybe because they can't handle stress. But if you're in your 20s, early 30s, not so likely. If you're in your early teens and after 30s, then again, the suicide risk goes up dramatically. We're not really sure why, and that's a great question. Maybe that's something that you might want to try addressing at some point. Okay, so I'm going to go through the next few slides. If you have any questions, please ask. This will be on your final exam. It's really important that you understand how we can go back and examine where these neurons are coming from, how we can understand what the processes are like, and again, in the context that if we can understand this fundamental process in the brain, maybe we can understand a whole host of different types of neurobiological phenomenon, ranging from mood disorders to neurodegenerative conditions, if this process impacts all of them. So within the dentate gyrus, in the subgranular zone layer of the dentate gyrus, so you'll have to imagine that sort of hook-like structure that I showed you a little bit earlier on, we have these stem cells. And these stem cells, again, they look a lot like radial glia. And they're described as radial glial-like stem cells. And these are the ones that exist in the subgranular zone layer. And these are the ones that undergo proliferation. They divide. And that's what a stem cell does. They're not really a neuron, they're not really a glia, they're just an amorphous stem cell that has the potential to become a number of other cells, and they proliferate and they renew their own population. And Nadia will come in and talk to you a little bit more about that later. I told you that the factor that involves this proliferation is really that VEGF. VEGF will enhance and accelerate this proliferation. So while you might have 4 here to begin with, that with VEGF you might have 8. It will enhance this proliferation greatly. I also told you that proliferation by itself means absolutely nothing. So you can have all of these proliferate and every last one of them dying. That doesn't do you any good at all. And so the survival of these proliferating cells is important, and that's the role of BDNF, where BDNF now comes in and promotes the survival of the ones that have just proliferated. And these immature adult-born neurons are important because these are the ones that are the most synaptically modifiable. They're the most plastic neurons that you will ever generate in your entire brain. Eventually, over the course of about 2 to 3 weeks, so this takes about a week's worth of time, 4 to 5 days-ish, about a week, And the rest of this process, the maturation, migration, synaptic incorporation, takes about 2 to 3 weeks for this to fully mature. So there are different time courses that are involved in this. So I wanted you to be aware of these different processes. Sorry, I'm just trying to coordinate with what's going on up there as well. I have too many of these. Okay, I think I'm back on track, and apologies on that. So this is going to be important, and I want you to be aware of this. At each of these different stages, so we start off with about a 4 to 5 day window, and then this 4 to 5 day window eventually leads to a few weeks worth of time. If I were to start here at time 0, let's assume that this is time 0, just before everything has begun, before this radioglial-like stem cell has started to divide, and in the presence of things like VEGF has started to proliferate, and then in the presence of BDNF to form these immature adult-born neurons in red, and before they eventually start migrating and incorporating into other parts of the hippocampus. If at time 0, I were to inject a chemical into my mice to be able to track dividing cells, I would inject a chemical like BRDU. And bromodeoxyuridine is incorporated into the nucleus and into the DNA of dividing cells. It only gets incorporated into dividing cells. Do you guys know, do neurons divide? Not mature neurons. In fact, let me ask you a trick question. Do neurons have the capacity to go through the cell cycle? Yes, at some point they must have, because they're differentiated. What stage of the cell cycle do neurons sit in? That's the trick question. Do you know? Sorry? Yes, that's right. Good. So neurons are actually sitting in this really weird phase of the cell cycle. They actually sit outside of the cell cycle, known as G0 or G0. And that means that they almost look like they're old cells, like they're senescent cells. And there's some idea that maybe if you drive them back into the cell cycle, they don't like it, and that's one of the reasons why they undergo apoptosis. Nevertheless, only these immature dividing stem cells are the ones that take up this BRDU. None of the other neurons in your brain, outside of the ones that are actually dividing like stem cells, will take up BRDU. Now, if we were to start staining all of these different cells, we could have a marker like Ki67, and in some places you'll see this as PCNA/Ki67. It's a proliferating cell nuclear antigen. It's a marker for proliferating cells, and you should know that it's a marker for proliferating cells. Once they have stopped proliferating and they start forming these immature neurons, we can no longer detect them using Ki67. At every stage, though, as long as it's an immature neuron or a stem cell, we can mark it with a protein known as double cortin. And double cortin is there right up until the time it actually becomes a mature neuron. So we can use double cortin, in other words, to mark proliferating cells, radioglial stem cells, and these immature neurons. But once they start forming synapses and they start forming calbindin, D28K, or calbindin CABP, these are a marker for mature neurons. I told you at the beginning, there's another marker that we're going to be seeing later on. Do you remember that marker at all? Nu-N would be another marker for these mature neurons as well. And so all of these processes take time, and we can track each stage at which these things are happening. And by doing these types of studies where you're marking for Ki67, where you're marking for double cortin, and to see the production of mature neurons, this is the way in which all of that material that I told you earlier on about stress and the effect on depression and the effect on neurogenesis was actually carried out. So again, you don't have to worry about all of these different diagrams. It's just to show you how you can go back and track these different molecules at these different stages. Not all of them are turned on all the time, but these are important to realize. I think actually now would be a good time to take a break before I actually get into the next group of these stem cells that are found in vivo. Before we get going, I had a couple of really quick questions to ask the class. So number one, how are you guys doing? Okay, good. Um, the second question is, we have an assignment that's due in about a week or so. How many of you have thought about that? A couple of you. I'm probably going to extend the deadline on that. I'm going to put up a couple of guidelines for you and then give you my own example as a continuing part of the project. It's relatively small in terms of the amount of work that you need to put into it. But the next part of the assignment is this future directions. I'm probably going to extend the deadline for about a week. We're not really sure what's going to happen with the looming TA strike. It doesn't really affect us too much here, to be quite honest. I do most of the grading anyways in terms of the midterm and everything else. However, one of the consequences of the TA strike is that if TAs decide to go on strike, and they're, that's completely up to them, that the other members of HMB would have to take up some of the slack in some of the other courses, which means that I might become like a de facto TA in someone else's class. We're not really sure how that's all going to be organized yet. And so I wanted to make sure that all of you were sort of on track, because I know things might get a little bit harried in terms of timelines, etc. So I'm probably going to push that back by about a week or so. Start thinking about future directions that you want to take in the paper that you've already started looking at, and what sorts of experiments you might do and some of the outcomes that you might be expecting. Yeah. Yes, it's exactly like proposing one or two follow-up experiments based on the results that were presented in the paper that you had. Like, what would you want to do next? It's very simple. Okay? Yes. Yes. Um, Potentially, like there is a makeup midterm tomorrow, and so we have to wait for the makeup midterm to be done before meeting. Yeah. It's a good question, but thank you for asking. Yes, another question. Yes. Absolutely. I will show you in my example. I will have a couple of other papers that I brought in that I think would be useful in the context of future directions. You should. You don't need to, but it would be beneficial if you did, because it will help your final assignment where you will be looking at other papers anyways. Yes. No, it's okay. Go ahead. Yeah. I'll try, but if you have a question, yeah. It's really not about giving a detailed plan, because that's really a grant proposal. What the future direction should be is a good rationale for why you would want to do those next. So, this was unresolved. It's still a big question about depression. We need to be able to address this, and this might be an experiment that might be proposed to answer that. And it's more about the rationale rather than telling me, I'm going to take like 20 mice, and then I'm going to do experiment A, and 20 mice and do experiment B. I'll compare them to controls, and then I'll get my answer. It's not about that. It's another good question, and hopefully I'll be able to address that some more later on. Okay, if there are no other questions, I'm just going to go on with this. One of the other areas that is that exhibits neurogenesis is this area of the brain known as the subventricular zone. So if this is the subventricular zone layer shown for you here in this brown, in this hashed gray color, one of the things that makes this unique is that some cells that are found in this subventricular zone on both sides in the adult will undergo proliferation as well as have stem cell-like features. And these areas include different cells like the ependymal cells and the subependymal cells that are shown in color around them. And one of the things that was known is if you take some of those cells from the subventricular zone, so you were to take this area of the brain and you were to scoop out this area of the brain, you were to make sure that you had individual cells based on all of the different cells that you took out of this region of this brain in the subventricular zone region, that some of those cells would have stem cell-like features, which would eventually become progenitor cells, and these progenitor cells in turn would, in theory, from this subventricular zone layer, be able to form different types of neurons, glia, etc. Now these neurons are actually very unique in the sense that these neurons normally from this subventricular zone region actually go on to form neurons in the olfactory bulb. And in fact, usually they form the olfactory bulb interneurons, or the inhibitory neurons in the olfactory bulb. So again, if you're looking at neurons that are being born here in this subventricular zone layer, they will eventually do the same process that I showed and outlined to you in the dentate gyrus. They have very, very similar mechanisms. They have very, very similar timelines. They go a lot farther though. Their migration pattern is actually much longer than in the hippocampus. They will go from this region, and if this is the mouse cortex, they go all the way out to the olfactory bulb, and they actually follow what's known as the RMS, or the rostral migratory stream. There is a set pathway for these newly dividing neurons to follow as they migrate toward the olfactory bulb, where they form these inhibitory interneurons in the olfactory bulb. And this is a continual process. Every single day, interneurons in the olfactory bulb die. They have to get replaced. New neurons from the subventricular zone proliferate, they start to migrate, and then they will differentiate once they reach this particular target. Some of you were asking during the break, are the markers that I showed you earlier on the only ones that are able to identify things like a proliferating cell, for example, a stem cell? I had mentioned GFAP was one of them, and I can't remember if I talked to you about nestin, but GFAP and nestin, for example, are ones that are typically used to identify things like stem cells. If they are these progenitor cells, we talked about double cortin, and nestin can also still be a marker here. In terms of as they migrate and as they start to form these more differentiated neurons, they'll still have that double cortin marker, just like we saw before, but they will also start to have other marker proteins like 2J1. And we won't go into the neural cell adhesion molecules at this point in time. I'll ask you to know that as they're migrating, these cells can also still express double cortin as well as 2J1. Okay, and we'll keep it really simple. At the end, before they begin their final differentiation process, they will still have that sort of immature state. They can still be marked by things like nu-N, but as they start to form mature neurons, we can start picking up other things like calbindin, D28K, and depending on the types of neurons they can be pushed toward, we can start seeing other markers of maturity like this marker here, tyrosine hydroxylase, and in inhibitory neurons, things like GAD65. Sorry, when I say GAD65, are you all okay with that? Have you learned that in your second year courses? You've heard of GAD, maybe? So do you remember tyrosine hydroxylase? Do you remember what that does? So that's the synthetic enzyme that will eventually form dopamine, right? So it's a marker of terminally differentiated dopaminergic neurons. We'll see that again today. GAD65 is a marker for terminally differentiated inhibitory neurons that contain GABA. So GAD65 is the glutamic acid decarboxylase enzyme that's involved as a marker protein for inhibition. And the other ones we won't really worry too much about right now. So this is one that I'd like you to make sure that you know. So there's, again, this is a really technically heavy lecture. But if I were to take a melon ball scooper, as Dr. Morshead likes to call it, and I scooped out the region in the subventricular zone and I put it down in a dish and I shook it apart and I got individual cells, one of the things that I would see is that each of those individual cells, if I put it into a tissue culture dish, would actually form what is a unique structure that is characteristic of stem cells, which is known as a neurosphere. So Stem cells like to adhere to each other. They like to stick together and clump together, a lot like neuroscience students. They like to kind of hang out together. And as a result, they form these balls in the neural culture dish. And these neurospheres are these structures that you see down here. And again, we won't worry about whether or not they're progenitors or true stem cells. That's more of a technical discussion that Nadia will have with you later. If you take this neurosphere, which again is a fundamental property of these stem cells that have been formed, and these form in the presence of things like epidermal growth factor, or EGF down here, and EFH that is shown. If you culture it in the presence of EFH, they start to proliferate and form these neurospheres, and also in the presence of that FGF, fibroblast growth factor, that we talked about when we looked at depression, as well as in the presence of heparin, that overall they would form these neurospheres. And if we kept it growing in this same medium, and we then dissociated, we took apart the neurospheres to single cells, and we took any one of those single cells and we did this whole process again, each of those cells in turn would start to form these neurospheres again. It's self-propagating. It can grow over and over again as long as you have these neurospheres in a dish. And you can keep them going for ages and ages, and they can keep going and dissociate them, pull them apart, take an individual cell, put it back into EFH, and each of those cells in turn will form these neurospheres. And you can keep it going, but if you wanted to test whether or not this had the capacity as true stem cells to form all the different cells in the nervous system, what you would do is add a substance known as fetal bovine serum, or FBS. And fetal bovine serum, no longer EFH, but fetal bovine serum, allows that stem cell to differentiate. And if you really had a stem cell that really could form every single cell type in your brain, you would actually be able to take a picture like this. They would actually start to, instead of growing as a ball or as a sphere, they would actually start to stick to the bottom of your dish. And as they stuck to the bottom of your dish, you would be able to stain for neurons, and you can use a marker like beta-3 tubulin. You could use NuN, for example, to see terminally differentiated neurons in the presence of FBS. You could stain for GFAP. Now this gets confusing, right? I told you earlier on, GFAP is a marker for stem cells, but in addition to that, it's also a marker for terminally differentiated astrocytes. And GFAP is a marker for both stem cells as well as for astrocytes that you see here in purple. The difference is that structurally they look very, very different. Again, another cell type that you have in your nervous system is these oligodendrocytes within your brain, and we can use an antibody against the O4 protein or glycoprotein that will stain those in this sort of pinkish red color. So do these original neurospheres form every last cell type in your brain? Yes, they do. And that's one of the reasons we're so interested in these. So in theory, if you get something from the subventricular zone and you are able to do so in terms of molecular and cellular biology, you could, in theory, start to form different types of specific neurons, maybe to replace neurons that are lost in Alzheimer's or in Parkinson's or other neurodegenerative conditions, maybe to form newly myelinating glial cells that might be important in central lesions associated with multiple sclerosis. And this is why stem cells are so important for us as neurobiologists. The last thing I'll talk about, and we're going to come back to this because it's something that as third-year neurobiology students and neuroscience students, I think that there's a lot of there's a lack of context around the whole physiology of exercise and the brain and its positive effects. And don't get me wrong, exercise has many, many positive effects on the brain and has many positive effects through BDNF. But I want you to actually understand the biology behind why we say exercise is good for things like depression, and it's good for things like increasing BDNF. It's good for cognition and everything else. Do you have a question at the back? If I heard your question correctly, Does the addition of FBS, or fetal bovine serum, induce the differentiation of the neurospheres into these differentiated states? Yes, it does. Okay, so some of those cells in those neurospheres will become neurons, some will become oligodendrocytes, and some will become these astrocytes. Yes? So actually, GFAP is a cytoskeletal marker. So it's an intermediate filament protein. Beta-3 tubulin is also not a surface marker, but it is a marker for the cytoskeleton. You have to permeabilize these cells. You have to allow the antibody to go into the cells, and that requires a detergent like Triton X100. Yep. It's a glycoprotein, so it's found both in the cytosol as well as on the cell surface. Yep. Any other questions before going on? Great technical questions. Don't need to worry about Triton permeabilization or anything else in terms of this particular slide. So if we go back and we were to take a look, I told you that one of the things that is important in this whole process, and one of the things that is important in things like depression, and I mentioned sort of as a bookend to the whole depression story, one of the things that I told you at the very outset was that when I was diagnosed with having depression, one of the things that I had a very tough choice in deciding was, based on the literature that I had been reading and based on my own knowledge and research about things, I had a choice of either going on antidepressant medication. I knew that it might work, it might not work, it would take some time. In addition to that, I really didn't want to go on antidepressant meds for other reasons. So I looked at the literature, and one of the things that I decided to do, even back 6 years ago, one of the things that was really important was exercise. And exercise, again, seems to be beneficial as well as somewhat protective against things like depression and other things. It wasn't really clear back then, like, why it's so protective. Why, for example, exercise might have led to a faster recovery, better recovery from depression. However, we now know that one of the things that exercise does is that it will increase BDNF. Now, right away, right off the top, you should be like screaming that, holy cow, what you're showing there is absolutely wrong. You just told us, for example, that VEGF is the thing that actually causes increased proliferation. BDNF just causes increased survival. But increased survival by itself is okay. And by the way, exercise also does induce VEGF in your brain as well. And that's something that gets completely forgotten. So through exercise, you can have a mouse or an exercising mouse, and all the panels kind of line up in this vertical region. And you can inject these mice with BRDU, that bromodioxyuridine that marks the DNA of dividing cells. And this is the structure that we looked at in the hippocampus. This is that dentate gyrus, that very characteristic sort of crescent shape structure in the hippocampus. And you can see that each of these little blobs that are marked in this dark color represents a nucleus that has been stained with BRDU. And it's showing proliferation occurs 12 days after BRDU injection. If you did the same thing in a regularly exercising mouse, one of the things that you would see is that in this regular exercising mouse, you would actually see increased proliferation. Originally, it was thought to be completely mediated by BDNF. We now know it's VEGF plus BDNF, because one of the things that seems to happen is that if you track all of these newly dividing neurons, all of these represent stem cells, in other words, 12 days after you've injected, if you follow it 6 weeks later, have most of these survived? No, they haven't. Most of those original BRDU labeled cells that marked the original dividing stem cells in the dentate gyrus at time 0, it doesn't mean that stem cells haven't continued. We just haven't marked them. But those ones that we did mark, most of them are gone. Right? So there's very little survival left in this non-exercising mouse. In the exercising mouse, we have increased rates of proliferation, not primarily due to BDNF, primarily due to VEGF actions. We have a lot more that you can see here. But if we follow these exercising mice out, you see that there is indeed an enhanced survival. And we'll come back to this role that BDNF proliferation, and many of these other factors have, not just in terms of mice, but in your neurobiology a little bit later on in the course. But I wanted you to be aware of all of the different controversies surrounding this. So I'm going to make a quick change over to the opposite. We've been talking about depression. We've been using depression sort of as a model system to understand different biological processes, things like proliferation of stem cells and how we can go and mark these different stem cells, animal models that might be important, and why we don't have a good model for really complex disorders like animal depression. The flip side of this is resilience. So resilience represents that your brain's capacity, or a mouse's brain's capacity, to cope with whatever environmental stress that it encounters. And in doing so, it's able to function. So again, if you ever go and see a mouse that has undergone social defeat or different forms of induced depression, they don't tend to function well. They don't tend to eat much. They don't tend to like drinking sugar water. They don't tend to move around as much either. And if you have resilience, you're able to function in the presence of this prolonged stress. In fact, there's a lot of research that's coming out now that if you were to actually engage in what's now known as active coping strategies, in which you perceive things like, one person might look at it as, and you all did this 2 weeks ago. Some of you did exactly this process, and it depends on how you had these active coping strategies. Some of you had like 4 midterms in a row, and some of you looked at it as, holy cow, this is like the worst thing that's ever happened to me, right? And some of you will have had different ways to cope with that, or different ways to perceive that, and some of you might have gone and said, at least I get them all out of the way and I can enjoy my reading week. And that's really the way in which these active coping strategies that allow you to perceive stressors in different ways, you can train yourself to get around this. And yes, it's psychological, but it also has a neurobiological basis. The other way that doesn't work, and it's not a good way to build up resilience, which again, psychologically and neurobiologically, which we'll examine next Tuesday, is to engage in what's known as avoidant coping. So you know that there's a stressor there. I'm just going to avoid that stressor even though it might follow me around. And that avoidance coping is actually the worst way to try to build up resilience. You will never be able to promote this behavioral adaptivity, nor will you gain any psychological resilience. It's really how you change your own internal perceptions. And I know it sounds purely psychological, but you're going to have to trust me that there is a biological basis that we're going to uncover next week on how resilience actually occurs in your brain as well as in animal model brains. So we want to, in this course, not look at the psychology as much. There are excellent psychology courses out there, by the way, that you should look at in terms of understanding these overall large mechanisms. They're really, really cool courses. They teach you a lot of different types of things. But in this course, we're really interested in the neurobiological basis and mechanisms involving resilience that protects you from stress. A lot of investigators have been really interested. As I told you earlier on, it's been known for a while. If you take genetically identical mice, for whatever reasons, maybe it's epigenetic, maybe there are other molecular factors. If you take genetically identical mice, like having a whole set of twins together, and you were to stress them out the same way, whether it's a forced swim test, whether it's chronic unpredictable stress, the majority, 2/3 of them to 3/4 of them, will actually have depression-like symptoms, but the other 1/3 to 1/4 are actually invulnerable to that stress. And if you breed them, they will all their offspring are also resilient to that form of stress as well. But the overall neurophysiological process, although the description of that in both animal literature as well as in human behavior has been well documented. You probably know individuals that have gone through a really, really rough time that would have wiped anyone else out, but they're perfectly fine. Like what causes them to be resilient? Is it really the coping mechanism? Have they learned how to cope with it? Or is there a change that has occurred within their brain? And this is sort of where we're heading. So in depression, as well as in resilience, there seems to be the involvement of this neural circuit that involves the reward system. And the reward system, again, if you think about anhedonia, I don't like doing things that I used to love doing. That's one of the signs, one of the principal cardinal signs of depression is this anhedonia. Anhedonia has to involve this reward system. This reward system is also intimately linked, for those of you who have taken other psychology courses, intimately linked to motivation as well, right? So you feel less motivated to do things, you don't like doing things that you used to before. As a result, you probably are going to be less active, etc. In this chronic social defeat model of depression that I mentioned to you at the very beginning, and I'll show you this slide in a few minutes, we see exactly the same thing. We see both susceptible and resilient. Some animals fall into depression, and some animals, no matter what other animal you put in with them, are resilient to falling into depression. And after 10 days of this, you can actually separate these two groups up. This group here will be resilient to social defeat stress. This group here is already showing signs of social defeat stress. Yes? That's a great question. In fact, it turns out that in terms of all the animal models that we look at, so we made this distinction between social defeat stress in terms of the VTA nucleus accumbens firing patterns versus the periaqueductal gray and delta phos B. But no matter what model you look at, there does seem to be a clear distinction between resilience as well as depression. Either one, you can see evidence of both. Yes? Resilience to stress in general that will, at least in animal models, cause depression or depressive-like behavior. Yes?
But they still have the same like firing. I still release neurotransmitters and like norepinephrine release. It's the same stress response, but it just doesn't have the
Again, a great question. Do they have the same stress response, so the same level of stress? Do they have the same level of glucocorticoid release and everything else? Or do they have less glucocorticoids, so they don't even get the same? So that's a great question. It turns out that they do, but the way in which their brain handles it is different, and we'll get into that next week. Is there another question out there? No? Okay, so I'm going to take you through the beginning of this paper. I'll take you through the full paper, and I'll put up the full paper for you. This paper actually comes in two different parts, and so I'll take you through the first part just to get you through some of the technical aspects before going through it in more detail. We talked about this learned helplessness, learned hopelessness model, some of these at the top, versus the social defeat model. And it's this social defeat model where the changes in this VTA firing rate seems to be more important versus the periaqueductal gray as well as the VTA changes in delta phos B. And we've already talked about that in terms of the molecular biology, but I want to really dissect this out in some more detail for you. In a number of different animal studies, and it's not just Eric Nestler's group, there are a number of different groups that have shown exactly the same thing. Multiple depressive symptoms in these susceptible mice have been implicated or linked to this hyperexcitability of the VTA group of neurons. And these VTA group of neurons are the ones that release dopamine. And this is that motivational reward circuit that I mentioned a few slides ago. One of the things that has been really, really hard in neurobiology over the last 50 years is a good way to be able to stimulate just the VTA neurons and no other neurons around it. It becomes really, really challenging. And so one of the ways in which scientists have started to get around this is by using sophisticated techniques like optogenetics. At U of T, there are a number of researchers that have started using optogenetics in their own labs and as part of their own research because of the power of the tool, of this particular tool. So optogenetic activation of specific groups of neurons within the VTA allow you to turn off this hyperactive group of neurons that are associated with depression or depressive-like behaviors, and only those neurons. Because you might argue before this had occurred that you are actually affecting a whole bunch. And I'm going to try to show you why we think that this is really important. So before I get there, I need to take you through some basic ideas and some concepts. Your VTA and nucleus accumbens is always firing. Your VTA and nucleus accumbens is always trying to figure out what's the best thing for me. And dopamine is sending that signal out to different areas of the brain, including the nucleus accumbens, trying to figure out, is this something that should be interesting to me? Should I be paying more attention? Should I like this, or should I completely avoid doing things like this? In the VTA, in that ventral tegmental area, these dopaminergic neurons actually show two basic firing patterns in vivo, in the normal state, in animals as well as in humans. And I'll show you that toward the end of the course. There is what's known as a low frequency, so the number of action potentials per second is rather slow. Tonic firing, and what do I mean by tonic firing? Any ideas? Yes. Yeah, it's always on. It's continually active, but it's always active at a very, very low basal rate, right? So your heart rate, you have a low tonic basal heart rate. It's always going, but you can speed it up simply by running across campus. And that would be like a high frequency phasic firing. So you have a basic heart rate, about 70 beats per second right now. That's that tonic state. But you can speed it up simply by running across campus, in which you're inducing this phasic activation. And it's exactly the same thing in the VTA. This phasic firing of the neurons in the VTA, though, is well known to encode for reward signals. But for whatever reasons, this gets inappropriately upregulated by repeated social defeat stress. So you get bullied by a larger animal, you get bullied by a large institution, you're going to have this social defeat stress. And it's been shown in this mouse model of depression that we get this overactivation, this phasic firing, inappropriate phasic firing of these VTA neurons. But it's only in those mice that actually succumb to social distress. If you have the same stressor that causes the same increase in glucocorticoids in another group of mice that don't fall into depression, those resilient mice actually see no change in the phasic firing pattern in their VTA. So again, it suggests that this might be one of the causes for why some individuals fall into depression versus being resilient. So if you don't overactivate your VTA in stressful situations, you don't get this phasic increase in these stressful situations. You have resilience versus having socially induced stress. So you're asking a really interesting question, and it gets into the fundamental basis of neurobiology that I don't have an answer for. So are you born with this, and can you learn this? Which I think is what you're asking. I was going to see if that was like a different effect on like VTA. Yeah. So I asked one of my colleagues the same question a couple of years ago, and I kind of agree with him. He's a psychiatrist. I'm more of a neurobiologist, and so I often ask a clinician these types of things. And he also believes that it's possible for you to train someone to become more resilient. And there's more literature coming out to suggest, both in terms of psychology and basic biological mechanisms, that you can actually train for greater resilience. It might not, you may never get to the same level as someone who has got a certain reserve resilience, but you can certainly do better and cope with stress better, and you can learn that resilience. And maybe that has to do with synaptic modifiability, but we're not really sure yet. Sorry, I have a lot of really unsatisfying answers in this course, but that's the best state of knowledge that I'm aware of. So one of the biggest issues is that had actually been known for about 5 years, 6 years now. The direct evidence that you can actually change things in real time. So imagine, in other words, I think you're depressed because you have an overactive VTA. And if I could turn that switch off right now, you will stop feeling depressed immediately, in real time. That's been incredibly difficult. If I could somehow do that, though, if I could somehow turn a switch on and off at will, in other words, I'd be able to prove without a doubt that resilience is an overactivation of this pathway versus another pathway. And that has been incredibly difficult. And scientists have been going back and forth trying to figure out, Is your experiment better than the experiments that have gone beforehand? But this new technique that I'm going to start unwrapping for you, optogenetics, actually allows you to start making this temporally and spatially precise. Precise enough that no one can really argue that you're not just affecting a whole group of neurons, you're affecting just the VTA neurons and no other neurons around it. You're not affecting glial cells, you're not affecting anything else. And so this is the topic of that 2013 paper that came out in Nature Medicine that I'll put up for you, and its companion papers that actually try to address, is the phasic firing pattern that is associated with depression versus a lack of a change in phasic firing in resilient individuals, can we indeed show this in real time? The way that I would have had to have done this if I was still in research many, many years ago, 10 years, 15 years ago. The way that anyone would have tried to have done this would be to have stuck in an electrode into an area of the brain like the VTA, and as a result of that, by putting in like an inhibitory current or inducing an electrical charge around a group of neurons, if I were targeting a specific neuronal type, I could change the rate-dependent response. I might see a therapeutic effect, but I might also activate other groups of neurons around it. It's not very sensitive, in other words. So I'm affecting a whole group of adjacent non-targeted neurons, even though I'm doing this with great precision. Like this electrode here is pretty small, but it's affecting much more of the neurons than I want in this cluster around it. So I'd never be able to say that I'm only affecting VTA. I might be affecting other neurons nearby, maybe in other targets that might be linked to motivation. And we'll get back to that again. So other studies have inadvertently, not because they wanted to, inadvertently activated other neurons through this electrical stimulation. You can also do lesion studies, scoop out whole groups of neurons, see the effect. If I scoop out that group of neurons, is it going to be equally effective? But those lesion studies are exactly the same. How do you know that you're only removing that group of neurons? And so you can't rule out any other side effects that might be associated with it. So you need a way to both temporally and spatially be very, very specific in terms of the neurons that you're turning on or turning off. And this modern approach is actually the one that seems to be the most efficacious for us right now. So traditional neuro has failed us in a lot of ways. We have poor spatial resolution, not just one group of neurons, probably many, many groups of neurons. Poor temporal resolution as well. The time course for certain techniques, if I were to turn off a group of neurons, I were injecting a pharmacological blocker, it might take upwards of 30 minutes. My behavior might actually occur in under 30 minutes. So I might not even be able to use a pharmacological manipulation. So there are both poor temporal and poor spatial resolutions. And again, lesion studies where I can inject a toxin, wipe out whole groups of neurons, also not very specific either. So this gets into this brand new field, relatively brand new, that started to work its way into the scientific literature back in 2009 and 2010. And it is one of the most highly adopted techniques that are now available for scientists. And I don't know if you've heard of optogenetics before, but I'm going to start telling you a little bit about different aspects of optogenetics. If you've gone to any of his talks, Carl Dijkstraat is actually a wonderful individual. If you have the opportunity to go and study elsewhere outside of Canada and you're really interested in neuroscience, the United States and Europe are the two places that you should be going to, because they're the places that will have these types of innovations that aren't really available to us now. If you could go to the lab at Stanford, for example, there are things that are in process and development there right now that aren't even in the papers yet that will completely amaze you. And so if you have the chance, you should go and try to join a lab like Carl Dijkstraat's lab at Stanford University or Ed Boyden's lab down at MIT. So what he did as a very young individual, he decided as a psychiatrist, he's a clinician scientist, he said, all this stuff that we've been doing in neuroscience is complete garbage. Like that electrical stimulation, I did that as a grad student, it's complete garbage. We need better, more refined techniques. And Carl Dijkstraat and Ed Boyden decided together that, you know, your your skull is both a detriment but also an advantage, in the sense that because of your skull, no light gets in to your neurons in your skull, and it's a dark enclosure. And so if I could find a way to turn on and on a light activatable switch in this dark space, I'd be able to actually activate certain groups of neurons. And that was sort of his line of reasoning. And so looking around in the scientific literature, one of the things that he found was that there were certain species of algae that were there that were responding and moving in response to certain certain wavelengths of light. And what he decided to do is that he wanted to take the cloned cation channels that were found in these different types of algae that were responsive to certain wavelengths of light that respond on the order of milliseconds, not minutes, not seconds, but on the order of milliseconds, that worked a lot like neuronal channels normally do in your brain. So they open and close really quickly, but instead of being opened and closed by a drug or by a neurotransmitter, they get turned on and off, open and close in response to flashes of light. And so this is what, again, Carl Diceroth, Ed Boyden, and a number of other colleagues, including Gottschalk, did in 2009. And their paper actually revolutionized neuroscience. Their first paper actually came out with this one protein known as channel rhodopsin 2. There are many newer modifications to channel rhodopsin 2, but in response to this blue fiber optic stimulation in certain neurons, that you are able to turn on and off groups of neurons in the order of milliseconds. They were able to turn on and off neurons simply by turning on and off this light, which is exactly what we would need to do to show that resilience occurs within the VTA. So this is excitation. I told you that those algae respond to many different wavelengths of light. And so the other cloning that occurred was for not the channel rhodopsins, but the halo rhodopsins and the other NPHR group of channels that you could also genetically engineer and deliver to cells that will turn off or inactivate neurons that express these channels. So these are generally assumed to be inhibitory. So these opsins, these channel rhodopsins, or these opsins, are natively expressed in a variety of different microorganisms, like algae. And using transgenic methods, this opsin gene can be introduced into whatever neurons you want, into whatever experimental animal you want, using stereotactic viral delivery methods. Okay. Let me just, sorry. Okay, so the other thing that makes this really cool, do neurons express channels? Yes. And so if neurons have the normal machinery to express channels, now you're delivering a different type of channel, but the machinery is already there. So you don't have to worry that it's a foreign gene that you're introducing that won't get expressed and targeted to the right regions. And so the internal machinery in your neurons are also amenable to this process as well. And this gets you again into different types of channel rhodopsins. Imagine I am delivering channel rhodopsin 2 or CHR2 that responds to blue light by opening up this non-selective cation, which would excite whatever neuron expresses this channel rhodopsin whenever blue light shone on it, versus some of those other halo rhodopsins like NP halo rhodopsin or archeo halo rhodopsin. These respond to longer wavelengths like orange and yellow flashes of light. And you can put these onto neurons, and when you express them in neurons using viral delivery, what you can do is, if this is the normal spiking pattern of this particular neuron, and you can see it generates an action potential. It goes from its resting membrane potential and spikes, and it spikes again. And now you want to see if it's spiking in relation to channel rhodopsin. If it's expressing channel rhodopsin, and you flash this blue light by this blue bar here, it will then spike and get into the same frequency as the flashes of light that go on to it. So it changes its firing pattern, in other words, to the firing pattern that is the flashes of light that are brought onto it. You can then also go in and do lots of interesting things to this neuron. You could express both channel rhodopsin as well as a halo rhodopsin. You can flash on blue light and orange light or red light. So again, you can flash on blue light, it will spike, another blue light, it will spike, and then in the presence of orange light, and if you spike with blue light, nothing happens. It inhibits this neuron completely. So you can turn on and off neurons completely at will on the order of milliseconds. Hopefully, you'll be able to piece together why we might want to do this in the VTA, especially in relation to resilience, because of the temporal and spatial specificity. Again, on the order of milliseconds, incredibly powerful, doesn't require anything in particular other than the ability to deliver genes. Unfortunately, I don't think this video is going to work on this particular system, but I will make sure that the video gets embedded for you on the digital resources page. Okay. I think I'm just going to go through this one or two next slides, and then we'll kind of wrap it up, and I'll take any questions that you have. I know it's been a really dense lecture in terms of molecular biology, but if you have any questions, I'll try my best to answer them. This is sort of an outline of what I'm going to take you through, hopefully a little bit today and more on Tuesday. These are the steps that you need to realize. All of these experiments now need to take place. So in other words, as a neurobiologist now, you have to be not only a good electrophysiologist, you also have to be a really good microscopist, because you'll see why you need microscopy in a moment. In addition to that, you also have to be pretty decent at molecular and cell biology, which is why hopefully you're gaining an appreciation of all of this now. So we know the genetic sequence of this opsin that is involved in being photoactivatable. You can turn on a light and it will open up a channel, and we know the genetic sequence. I told you that we need spatial resolution. We need to target specific types of neurons. In order to do that, I need to be able to have some genetic identity to where this gene is going to be expressed. So I only want it expressed in dopaminergic neurons. I have to know a little bit about the promoter that is found only in dopaminergic neurons, or only in excitatory neurons, or only in astrocytes. Whatever it is, I need to know some of the promoters that drive genetic expression. In addition to that, you also have to take into account, and we talked about this very briefly earlier on, you can infect neurons. Some of you already have infected neurons. Some of your infected neurons still carry the virus, and that's because neurons can get easily infected. And so in order for us to deliver this genetic package in step one to our neurons, the best way to deliver that genetic package is to deliver it through a virus. Viruses infect neurons extremely well. And so we need to take into account different types of viruses that we package this genetic construct into. And that's step two. Again, technically really challenging. The next step is, once you have this virus, you have to be a good surgeon in addition to being a good geneticist, a good molecular biologist, and now you have to be good at stereotactic surgery. You have to be able to deliver this to a precise area of the brain where you find that group of neurons, into the VTA, for example, or in other areas. And again, using that same surgical procedure, you have to be able to implant a fiber optic cable. Nowadays, we use wireless fiber optics, so you don't need to hook it up to a box. You can just have it sort of as a little stump on top of the animal's head. But previously, you needed an entire fiber optic cable. And then you would be able to go in and start measuring the behavioral responses, again, meaning you have to know some psychology as well as behavioral biology in order to measure the output of what you're measuring. So all of these things are going to be elements that I'm going to start unwrapping for you. So this is the stereotactic part of it. You don't have to worry about all of the details here. You don't need to memorize any of this at all. How many of you have done stereotactic surgery? One person. Okay. Anyone? Two. Okay. For those of you who have done stereotactic surgery, you know that this isn't the way we do stereotactic surgery. You actually have to immobilize the head. There are guide bars that make sure that the head is completely affixed, and you can measure certain landmarks. if you have the opportunity, it's something that is a very useful technique in neurobiology for all of you to learn. I happen to be really lucky. I got taught by an orthopedic surgeon, and as an orthopedic surgeon, as a graduate student learning from him, it was great because he taught me precise surgical techniques. And I remember doing my very first stereotactic injection by myself. He wasn't kind of helping me out with this. And by that time, I was really, really cocky. And so as I was brushing my long, flowing locks away from my head and tracking him down, I said, Paul, you got to go check out my surgery. It's fantastic, man. It's better than yours. So Paul was an orthopedic surgeon. He comes over and he goes, see, Paul, look, absolutely no bleeding. Like when you do your surgery, at least you have some bleeding and everything, right? Like mine, perfect, no bleeding at all. And I'm kind of like, again, brushing my long locks from my head and kind of waiting for him to say something great. He says, Bill, this is a fantastic surgery. Were you expecting your animal to survive? And I said, yeah, of course I was. And he said, your animal's been dead for about the last hour, which is why there's no bleeding. So this is a good reason why you should learn surgery from really, really good people, and to understand like some of the physiology that you learn in class is actually really important to you. So, you know, check to see if your animal is breathing from time to time. So in any case, there are many, many steps that are involved in this process. Eventually, you'll be delivering your virus a very, very small amount. 2 microliters might sound like it's a really small amount, but in the closed environment of the brain, this volume is actually enough to cause pneumotactic cell death in that area. And we'll talk about what gets delivered there in the next lecture. So if you have any questions, now's the time to ask. It was a really technically dense lecture. The next lecture will also be technically dense. Then we move into March, where we have some more guest speakers coming in, more of a clinical basis for the types of things that we look at here. Is that we actually still have room for one more guest speaker. And I'm kind of looking around at what you might, as a class, want to hear about. Is there anything clinical that you would like to hear about as a class? Sorry? Resilience. Okay, I could invite someone in to talk to you about psychiatric resilience. Anything else that you might be interested in case I can't get him? Yes. Autism or ADHD? Okay, so we can look into a clinician that deals with pediatric neurology. Okay, great. We do have a neurologist coming in to talk to us about Parkinson's. We'll learn a little bit about epigenetics and its effects on neurological behaviors, and we'll try to get one more guest speaker in as well. Did you have another person, Jacqueline? Focused ultrasound, disruption of the blood-brain barrier and delivery of therapeutic molecules? Yeah, maybe. Okay. We don't really talk about the blood-brain barrier in this course. I think you talk about it in 320 a little bit. So maybe. Thanks for the great suggestions. Have a great weekend. If you need to see me, I'm around tomorrow most of the day until about 4 o'clock, and you're just free to drop in.